Topic: Molecules and their interaction relevant to biology. Subtopic A: Structure of atom. The structure of an atom forms the fundamental basis for understanding all molecular interactions in biological systems. An atom comprises a dense nucleus containing positively charged protons and neutrally charged neutrons, surrounded by negatively charged electrons in orbitals. The number of protons defines the atomic number, whereas the sum of protons and neutrons gives the mass number. Biological processes rely heavily on elements such as hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur. These atoms interact through chemical bonding, and the behavior of electrons in orbitals is key to understanding these interactions. Electrons are arranged in discrete energy levels, shells, and subshells s, p, d, f. Each orbital can hold a specific number of electrons, s2p, 6d, 10, and f14. The electronic configuration follows the Aufbau principle, Pauli's exclusion principle, and Hund's rule, ensuring stability through optimal distribution of electrons. In biological molecules, valence electrons, those in the outermost shell, are most crucial, as they determine the atom's ability to form chemical bonds, such as covalent, ionic, and hydrogen bonds, which underlie all biomolecular structures. The quantum mechanical model of the atom is central to biology. According to this model, electron positions are not fixed but exist in orbitals with defined probability distributions. Quantum numbers, principal N, azimuthal L, magnetic ML, and spin ms define the energy, shape, and orientation of these orbitals. Biological specificity arises from the three-dimensional arrangements of atoms in molecules, which are determined by the shape and hybridization of orbitals. Isotopes of elements, which differ in neutron number but not proton number, have vital biological applications. For instance, radioisotopes such as cubed H, to the power of 4C, and cube squared P are used in labeling experiments to track molecular pathways, while stable isotopes are used in metabolic flux analysis. Understanding atomic structure is also essential in spectroscopic techniques like NMR and ESR, where atomic nuclei or unpaired electrons respond to magnetic fields, revealing molecular structures. The behavior of electrons underpins chemical reactivity, bond formation, and molecular shape, which in turn determines function in biomolecules. For example, carbon's ability to form 4 covalent bonds through sp³ hybridization allows complex molecular backbones in proteins. The behavior of electrons underpins chemical reactivity, bond formation, and molecular shape, which in turn determines function in biomolecules. For example, carbon's ability to form 4 covalent bonds through sp³ hybridization allows complex molecular backbones in proteins, DNA, and lipids. Electronegative elements like oxygen and nitrogen form polar bonds, enabling hydrogen bonding, which is central to the structure of DNA, proteins, and enzymes. Moreover, redox reactions, central to cellular respiration and photosynthesis, involve the transfer of electrons between atoms. Understanding which atoms are likely to donate or accept electrons based on electronegativity and ionization energy helps explain key pathways such as the electron transport chain. At the core of such mechanisms is the concept of atoms and their electron configurations. In essence, the structure of atoms provides the blueprint for all biochemical interactions. From the double helix of DNA to the folding of proteins and the specificity of enzyme-substrate binding, every interaction is rooted in the principles of atomic structure. A strong foundation in atomic theory is therefore indispensable for mastering molecular biology. Previous year questions PYQs with answers 1. CSIR Net June 2015. Which of the following statements about atomic orbitals is correct? A. The 2p orbital can hold up to 6 electrons. B. The 2s orbital has a higher energy than the 2p orbital. C. Electrons in the same orbital must have the same spin. D. The shape of the 2p orbital is spherical. Answer. A. The 2p orbital can hold up to 6 electrons. 2. CSIR Net Deck 2018. Which set of quantum numbers is not allowed for any electron in an atom? A. N equals 2, L equals 1, ML equals 0, Ms equals plus 1/2. B. N equals 1, L equals 0, ML equals 0, Ms equals minus 1/2. C. N equals 3, L equals 3, ML equals minus 2, Ms equals plus 1/2. D. N equals 4, L equals 2, ML equals 2, Ms equals minus 1/2. Answer. C. N equals 3, L equals 3, ML equals minus 2, Ms equals plus 1/2. L must be an LT, N, so L equals 3 is invalid for N equals 3. 3. CSIR Net June 2012. Which of the following explains the maximum number of electrons in a d orbital? A. 4. B. 6. C. 10. D. 14. Answer. C. 10. Would you like me to proceed with the next subtopic, like chemical bonds and forces that stabilize biomolecules, or any other topic from this unit? You said. Molecules. ChatGPT said. Excellent. Let's now move to the next topic. CSIR Net JRF Life Sciences High Level Lecture Unit, Molecules and their interaction relevant to biology. Topic B. Molecules, types and importance in biology. In biology, molecules are the physical substance through which life processes operate. They range from small inorganic compounds like water and salts to complex organic macromolecules such as proteins, nucleic acids, carbohydrates, and lipids. The foundational difference between living and non-living matter lies not in the type of atoms involved, but in the arrangement and interaction of molecules. All biological molecules are composed primarily of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, often referred to as the CHONPS elements. Inorganic molecules, such as water HO, are crucial due to their unique polarity and hydrogen bonding capabilities. Water's high specific heat, surface tension, and cohesive properties make it an ideal solvent for biochemical reactions. Ions like Na, K, Ca squared, Cl, and Mg squared serve vital roles in nerve conduction, muscle contraction, enzyme activity, and osmotic balance. For instance, Ca squared acts as a secondary messenger in signal transduction, while H concentration pH influences enzyme kinetics and protein stability. Organic molecules form the backbone of life. They are classified into 4 major groups. Carbohydrates are the primary source of energy and structural components. Monosaccharides like glucose serve as quick energy sources, while polysaccharides like cellulose and glycogen serve structural and storage roles, respectively. Proteins, composed of amino acids, are the most functionally diverse biomolecules. Their primary, secondary, tertiary, and quaternary structures dictate their roles in enzymatic catalysis, transport, signaling, and structural integrity. The nature of side chains determines hydrophobicity, charge, and interaction with other molecules, essential for protein folding and function. Lipids are hydrophobic molecules involved in membrane structure, energy storage, and signaling. Phospholipids form the bilayer of biological membranes. Cholesterol modulates membrane fluidity. Steroid hormones, derived from lipids, are essential for intercellular communication. Nucleic acids DNA and RNA are carriers of genetic information. Composed of nucleotides, they possess the ability to store, replicate, and express genetic information. ATP, a nucleotide derivative, acts as the universal energy currency of the cell. In biological systems, macromolecules do not function in isolation. They interact dynamically, forming molecular complexes, enzyme-substrate interactions, signal transduction cascades, and genetic regulatory networks. These interactions are governed by non-covalent forces such as hydrogen bonding, ionic interactions, hydrophobic effects, and van der Waals forces, making them reversible and specific, features essential for life. The three-dimensional structure of molecules determines their biological activity. For example, enzyme-substrate specificity is a direct outcome of molecular shape and charge complementarity. Similarly, the double helical structure of DNA arises from specific base pairing and hydrogen bonding between complementary nucleotides, stabilized by stacking interactions. Biologically important molecules are also sensitive to environmental conditions such as temperature, pH, and ionic strength, which can alter their conformation and functionality. Understanding molecular behavior under these conditions is crucial in biochemistry and molecular biology experiments, including PCR, enzyme assays, and electrophoresis. Lastly, molecular evolution examines how biological molecules evolve over time, providing insights into the origin of life, species divergence, and adaptation. Molecular evolution examines how biological molecules evolve over time, providing insights into the origin of life, species divergence, and adaptation. Comparative analysis of molecular sequences, DNA, RNA, protein has become a cornerstone in phylogenetics and evolutionary biology. In summary, molecules are the architects and operators of life. Their structure, function, and interaction are at the heart of all biological processes, from metabolism to heredity, and their study forms the cornerstone of modern life sciences. Previous year questions PYQs with answers 1. CSIR Net Deck 2013. Which of the following macromolecules shows maximum diversity in structure and function? A. Carbohydrates. B. Lipids. C. Proteins. D. Nucleic acids. Answer. C. Proteins. 2. CSIR Net June 2017. Which one of the following properties of water makes it suitable as a biological solvent? A. Low polarity. B. Low heat capacity. C. High dielectric constant. D. Low surface tension. Answer. C. High dielectric constant. 3. CSIR Net December 2015. Which one of the following molecules acts as both an energy currency and a nucleic acid building block? A. NAD. B. ATPC. GTPD. ADP. Answer. B. ATP. Would you like to continue with the next topic, such as chemical bonds and forces stabilizing biomolecules, or a new unit like cell biology or gene expression? You said, chemical bonds. ChatGPT said. Here is your CSIR Net JRF Life Sciences detailed lecture on which of the following macromolecules shows maximum diversity in structure and function? A. Carbohydrates. B. Lipids. C. Proteins. D. Nucleic acids. Answer. C. Proteins. 2. CSIR Net June 2017. Which one of the following properties of water makes it suitable as a biological solvent? A. Low polarity. B. Low heat capacity. C. High dielectric constant. D. Low surface tension. Answer. C. High dielectric constant. 3. CSIR Net December 2015. Which one of the following molecules acts as both an energy currency and a nucleic acid building block? A. NAD. B. ATPC. GTPD. ADP. Answer. B. ATP. Would you like to continue with the next topic, such as chemical bonds and forces stabilizing biomolecules, or a new unit like cell biology or gene expression? You said, chemical bonds. ChatGPT said. Here is your CSIR Net JRF Life Sciences detailed lecture on Unit, molecules and their interaction relevant to biology. Topic C, chemical bonds and forces that stabilize biomolecules. Chemical bonds and intermolecular forces form the backbone of molecular interactions in biological systems. Understanding these interactions is critical to interpreting how molecules acquire structure, stability, and function inside the cell. These bonds not only determine molecular formation but also mediate protein folding, DNA stability, enzyme-substrate interaction, cell membrane architecture, and signal transduction. At the core are strong covalent bonds, which form when atoms share electrons to complete their valence shells. Covalent bonds are the primary bonds that link atoms within biomolecules. For instance, peptide bonds link amino acids in proteins, and phosphodiester bonds connect nucleotides in DNA and RNA. These bonds have high bond energies, typically 80 to 100 kcal per mole, making them stable under physiological conditions. Ionic bonds, electrostatic interactions, occur between atoms with opposite charges, such as between a cation, e.g., Na, and an anion, e.g., Cl. In biological macromolecules, ionic bonds form between charged side chains of amino acids, like lysine positive and glutamate negative, and are particularly strong in hydrophobic environments like protein interiors. However, in aqueous environments, their strength decreases due to solvation by water molecules and the high dielectric constant of water. Hydrogen bonds are vital in stabilizing secondary and tertiary structures of proteins and the double helical structure of DNA. These bonds form when a hydrogen atom covalently bonded to an electronegative atom, like N or O, interacts with another electronegative atom. Though individually weak, approximately 1 to 5 kcal per mole, their directionality and multiplicity make them powerful when collectively present. For example, the Watson-Crick base pairing in DNA is held together by specific hydrogen bonds, At with 2, Gc with 3. Van der Waals forces are weak interactions caused by transient dipole moments when electron clouds of two non-bonded atoms fluctuate and induce mutual polarization. These forces play an essential role in molecular packing, such as in the hydrophobic cores of proteins, where atoms are packed tightly for stability. Though individually feeble, collectively they contribute significantly to protein folding and molecular recognition. Hydrophobic interactions are not true bonds but entropically driven forces where nonpolar molecules or parts of molecules aggregate to exclude water molecules. This is crucial in the formation of lipid bilayers, membrane proteins, and protein folding, where nonpolar side chains collapse into the protein's interior to minimize exposure to aqueous surroundings. These interactions are strengthened by the entropy gain from releasing ordered water molecules that otherwise form cages around nonpolar surfaces. Disulfide bonds, formed by the oxidation of two cysteine residues, are covalent but unique to proteins, especially extracellular ones. These stabilize tertiary and quaternary structures, particularly in secreted proteins, antibodies, and insulin. All these forces collectively determine the conformation and stability of biomolecules. For example, in proteins, the alpha helix and beta sheet structures are stabilized by hydrogen bonds, while the overall 3D shape arises from a combination of hydrogen bonding, hydrophobic interactions, van der Waals forces, ionic interactions, and occasionally disulfide bridges. The specificity and reversibility of weak interactions make them ideal for dynamic biological processes such as enzyme-substrate binding, antigen-antibody recognition, and hormone-receptor interactions, where transient binding and unbinding are essential. In DNA-protein interactions, such as in transcription factors binding to promoter regions, electrostatic attraction, hydrogen bonding, and shape complementarity all contribute to specificity. Similarly, in signal transduction, ligand binding to receptors involves multiple weak bonds to ensure reversible, regulated interactions. In conclusion, while covalent bonds provide stability and permanence in molecular structures, non-covalent interactions provide the flexibility, specificity, and dynamic behavior necessary for life. The cooperative action of these forces ensures that biomolecules are stable yet adaptable, a balance that is crucial for biological function. Previous year questions PYQs with answers 1. CSIR Net June 2016. Hydrophobic interactions are primarily driven by a formation of hydrogen bonds between hydrophobic groups. B. Dipole-dipole attraction. C. Entropy-driven exclusion of water. D. Covalent bonding between hydrophobic groups. Answer. C. Entropy-driven exclusion of water. 2. CSIR Net Deck 2019. Which of the following bonds interactions is strongest under physiological conditions? A. Hydrogen bond. B. Ionic bond. C. Van der Waals interaction. D. Covalent bond. Answer. D. Covalent bond. 3. CSIR Net June 2014. In the alpha helix structure of proteins, which type of bond stabilizes the helical turn? A. Ionic bond. B. Disulfide bond. C. Hydrogen bond. D. Peptide bond. Answer. C. Hydrogen bond. Would you like me to continue with the next topic, such as composition, structure and chemical bonds and intermolecular forces form the backbone of molecular interactions in biological systems. Understanding these interactions is critical to interpreting how molecules acquire structure, stability, and function inside the cell. These bonds not only determine molecular formation but also mediate protein folding, DNA stability, enzyme-substrate interaction, cell membrane architecture, and signal transduction. At the core are strong covalent bonds, which form when atoms share electrons to complete their valence shells. Covalent bonds are the primary bonds that link atoms within biomolecules. For instance, peptide bonds link amino acids in proteins, and phosphodiester bonds connect nucleotides in DNA and RNA. These bonds have high bond energies, typically 80 to 100 kcal per mole, making them stable under physiological conditions. Ionic bonds, electrostatic interactions, occur between atoms with opposite charges, such as between a cation, e.g., Na, and an anion, e.g., Cl. In biological macromolecules, ionic bonds form between charged side chains of amino acids, like lysine positive and glutamate negative, and are particularly strong in hydrophobic environments like protein interiors. However, in aqueous environments, their strength decreases due to solvation by water molecules and the high dielectric constant of water. Hydrogen bonds are vital in stabilizing secondary and tertiary structures of proteins and the double helical structure of DNA. These bonds form when a hydrogen atom covalently bonded to an electronegative atom, like N or O, interacts with another electronegative atom. Though individually weak, approximately 1 to 5 kcal per mole, their directionality and multiplicity make them powerful when collectively present. For example, the Watson-Crick base pairing in DNA is held together by specific hydrogen bonds, AT with 2, GC with 3. Van der Waals forces are weak interactions caused by transient dipole moments when electron clouds of two non-bonded atoms fluctuate and induce mutual polarization. These forces play an essential role in molecular packing, such as in the hydrophobic cores of proteins, where atoms are packed tightly for stability. Though individually feeble, collectively they contribute significantly to protein folding and molecular recognition. Hydrophobic interactions are not true bonds but entropically driven forces where nonpolar molecules or parts of molecules aggregate to exclude water molecules. This is crucial in the formation of lipid bilayers, membrane proteins, and protein folding, where nonpolar side chains collapse into the protein's interior to minimize exposure to aqueous surroundings. These interactions are strengthened by the entropy gain from releasing ordered water molecules that otherwise form cages around nonpolar surfaces. Disulfide bonds, formed by the oxidation of two cysteine residues, are covalent but unique to proteins, especially extracellular ones. These stabilize tertiary and quaternary structures, particularly in secreted proteins, antibodies, and insulin. All these forces collectively determine the conformation and stability of biomolecules. For example, in proteins, the alpha helix and beta sheet structures are stabilized by hydrogen bonds, while the overall 3D shape arises from a combination of hydrogen bonding, hydrophobic interactions, van der Waals forces, ionic interactions, and occasionally disulfide bridges. The specificity and reversibility of weak interactions make them ideal for dynamic biological processes such as enzyme-substrate binding, antigen-antibody recognition, and hormone-receptor interactions, where transient binding and unbinding are essential. In DNA-protein interactions, such as in transcription factors binding to promoter regions, electrostatic attraction, hydrogen bonding, and shape complementarity all contribute to specificity. Similarly, in signal transduction, ligand binding to receptors involves multiple weak bonds to ensure reversible, regulated interactions. In conclusion, while covalent bonds provide stability and permanence in molecular structures, non-covalent interactions provide the flexibility, specificity, and dynamic behavior necessary for life. The cooperative action of these forces ensures that biomolecules are stable yet adaptable, a balance that is crucial for biological function. Previous year questions PYQs with answers 1. CSIR Net June 2016. Hydrophobic interactions are primarily driven by a formation of hydrogen bonds between hydrophobic groups. B. Dipole-dipole attraction. C. Entropy-driven exclusion of water. D. Covalent bonding between hydrophobic groups. Answer. C. Entropy-driven exclusion of water. 2. CSIR Net Deck 2019. Which of the following bonds interactions is strongest under physiological conditions? A. Hydrogen bond. B. Ionic bond. C. Van der Waals interaction. D. Covalent bond. Answer. D. Covalent bond. 3. CSIR Net June 2014. In the alpha helix structure of proteins, which type? In the alpha helix structure of proteins, which type of bond stabilizes the helical turn? A. Ionic bond. B. Disulfide bond. C. Hydrogen bond. D. Peptide bond. Answer. C. Hydrogen bond. Would you like me to continue with the next topic, such as composition, structure and function of biomolecules, or move to another unit like cell biology or gene expression? You said. Composition, structure and function of biomolecules. Carbohydrates. ChatGPT said. Here is your CSIR Net JRF Life Sciences high quality lecture on unit, molecules and their interaction relevant to biology. Topic D. Composition, structure and function of biomolecules. Carbohydrates. Carbohydrates are one of the primary classes of biomolecules that serve as energy sources, structural materials, and recognition molecules in biological systems. Chemically, carbohydrates are defined as polyhydroxyaldehydes or ketones and their derivatives. They are composed of carbon C, hydrogen H, and oxygen O, usually in the empirical formula CHON. Their structural diversity arises from variations in chain length, functional groups, glycosidic linkages, and branching patterns. Carbohydrates are classified into three main types. Monosaccharides, simple sugars e.g., glucose, fructose, galactose. Oligosaccharides, short chains of monosaccharide units e.g., sucrose, lactose. Polysaccharides, long chains of monosaccharides e.g., starch, glycogen, cellulose. Monosaccharides. Monosaccharides are the basic building blocks of carbohydrates. They are classified based on the number of carbon atoms triose, tetrose, pentose, hexose, and the type of carbonyl group aldose or ketose. For example, glucose and aldohexose and fructose, a ketohexose, are biologically important sugars. Monosaccharides exist in linear and cyclic forms, with the cyclic forms predominating in solution. The cyclic structure forms due to an internal hemiacetal in aldoses or hemiketal in ketoses reaction, creating furanose 5-membered or pyranose 6-membered rings. Glucose predominantly exists in the pyranose form, and the anomeric carbon can form alpha or beta anomers, depending on the orientation of the hydroxyl group. Oligosaccharides. Oligosaccharides are formed by glycosidic bonds between monosaccharide units. A glycosidic bond is a covalent bond formed between the anomeric carbon of one sugar and a hydroxyl group of another. The disaccharide sucrose is composed of glucose and fructose linked via an alpha 1 right pointing arrow beta 2 glycosidic bond. Lactose, found in milk, is a beta 1 right pointing arrow 4 linkage between galactose and glucose. Oligosaccharides are often covalently attached to lipids, glycolipids, or proteins, glycoproteins, on the cell surface, where they play crucial roles in cell recognition, immune responses, and pathogen-host interactions. Polysaccharides. Polysaccharides are long chains of monosaccharide residues, either homopolysaccharides, same sugar units, or heteropolysaccharides, different sugar units. Storage polysaccharides. Starch plants composed of amylose linear alpha 1 right pointing arrow 4 linkages, and amylopectin branched with alpha 1 right pointing arrow 6 linkages. Glycogen animals, similar to amylopectin but more highly branched. These polysaccharides are water-soluble, compact, and rapidly mobilizable for energy. Structural polysaccharides. Cellulose plants linear chains of beta 1 right pointing arrow 4 linked glucose, forms rigid microfibrils due to extensive hydrogen bonding. Chitin arthropods, fungi composed of N-acetylglucosamine units linked by beta 1 right pointing arrow 4 bonds, forming strong exoskeletons. Unlike storage polysaccharides, structural polysaccharides are insoluble and resistant to digestion due to their beta linkages. Functions of carbohydrates. Energy source. Glucose is the central sugar in energy metabolism. Its oxidation via glycolysis and the citric acid cycle generates ATP. Energy storage. Starch and glycogen act as reservoirs of glucose in plants and animals, respectively. Structural role. Cellulose in plants and chitin in arthropods provide mechanical strength and rigidity. Cell communication and immunity. Glycoproteins and glycolipids on cell surfaces are involved in signal transduction, cell-cell adhesion, and immune responses. Precursor molecules. Carbohydrates serve as precursors for nucleotides, ribose in RNA and deoxyribose in DNA, cofactors, NAD, FAD, and amino acids. Stereochemistry and isomerism. Monosaccharides exhibit stereoisomerism due to the presence of chiral carbons. For instance, D and L forms refer to the orientation of the hydroxyl group on the chiral carbon farthest from the carbonyl group. In nature, D sugars are predominant. Apomers are sugars that differ in configuration at one chiral center, e.g., glucose and galactose differ at C4. Significance in research and medicine. Carbohydrates are key in biotechnology, glycomics, and medicine. For example, blood group antigens A, B, AB, O are determined by specific carbohydrate residues on red blood cells. In microbial pathogenesis, carbohydrate moieties are used for host attachment. Lectins, which bind specific sugar residues, are used in biochemical assays and drug targeting. Previous year questions PYQs with answers 1. CSIR Net June 2015. Which of the following linkages is present in cellulose? A. Alpha 1 right pointing arrow 4. B. Alpha 1 right pointing arrow 6. C. Beta 1 right pointing arrow 4. D. Beta 1 right pointing arrow 6. Answer. C. Beta 1 right pointing arrow 4. 2. CSIR Net Deck 2018. Which of the following is a branched polymer of glucose in animals? A. Amylose. B. Glycogen. C. Cellulose. D. Chitin. Answer. B. Glycogen. 3. CSIR Net June 2013. Lactose is made up of which two monosaccharides? A. Glucose and fructose. B. Glucose and galactose. C. Glucose and mannose. D. Glucose and glucose. Answer. B. Glucose and galactose. Let me know if you want the next topic, lipids, proteins, or nucleic acids, in the same detailed format. You said, lipids, ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on Unit, molecules and their interaction relevant to biology. Topic E, composition, structure and function of biomolecules, lipids. Lipids are a diverse group of hydrophobic or amphipathic organic molecules that are insoluble in water but soluble in nonpolar solvents like chloroform, benzene, or ether. They play crucial roles in energy storage, membrane structure, signaling, and insulation. Unlike carbohydrates and proteins, lipids are not true polymers, as they are not composed of repeating monomeric units in a strict sense, yet they form large macromolecular assemblies essential for life. Lipids are broadly categorized into simple lipids, compound lipids, and derived lipids, depending on their structure and function. 1. Simple lipids. Simple lipids include fats, triglycerides, and waxes. Fats are esters of fatty acids with glycerol. A triglyceride contains 3 fatty acids esterified to a glycerol backbone. Fatty acids may be saturated, no double bonds, e.g., palmitic acid, stearic acid. Unsaturated, 1 or more double bonds, e.g., oleic acid, linoleic acid. The degree of saturation affects melting point and fluidity. Saturated fats are solid at room temperature, while unsaturated fats are liquid. Waxes are esters of long-chain fatty acids with long-chain alcohols. They form protective coatings in plants and animals, e.g., cumin in plants, beeswax. 2. Compound lipids. These are lipids containing other elements like phosphorus, nitrogen, or carbohydrates. They include A. Phospholipids. Composed of glycerol, 2 fatty acids, and a phosphate group with a polar head, e.g., choline, ethanolamine. They are amphipathic and form the lipid bilayer of cell membranes. Types include phosphoglycerides, e.g., phosphatidylcholine. Sphingolipids contain sphingosine instead of glycerol. B. Glycolipids. Composed of lipid molecules covalently bonded to carbohydrates. They are predominantly found on the extracellular surface of membranes and are involved in cell-cell recognition and immunological functions. C. Lipoproteins. These are complexes of lipids and proteins responsible for the transport of lipids in blood. They are classified based on density, HDL, LDL, VLDL, kilomicrons. High LDL levels are linked to cardiovascular diseases. 3. Derived lipids. These are products derived from hydrolysis of simple and compound lipids. They include steroids, 4 fused rings, cyclopentanoperhydrophenanthran nucleus. Examples include cholesterol, testosterone, estradiol, and cortisol. Cholesterol is an essential component of membranes and a precursor of steroid hormones and bile acids. Eicosanoids, derived from arachidonic acid, include prostaglandins, thromboxanes, leukotrienes. They are involved in inflammation, immunity, blood clotting, and smooth muscle contraction. Fat-soluble vitamins, A, D, E, K. These are isoprenoid derivatives essential for various physiological functions such as vision, vitamin A, calcium metabolism, vitamin D, antioxidation, vitamin E, and blood clotting, vitamin K. Biological functions of lipids. Energy storage. Lipids yield twice as much energy per gram compared to carbohydrates or proteins. Triacylglycerols are stored in adipose tissue and mobilized during fasting. Structural role. Phospholipids and cholesterol are fundamental components of biological membranes, regulating fluidity and permeability. Signaling. Lipids act as second messengers, e.g., diacylglycerol, IP, and hormones, e.g., steroid hormones. Insulation and protection. Fat layers insulate body temperature and protect organs against mechanical shock. Absorption of fat-soluble vitamins. Lipids are necessary for the digestion and absorption of vitamins A, D, E, and K. Fatty acids, saturation, chain length, and function. Fatty acids are key components of lipids, varying in chain length, short, medium, long, and degree of saturation. Essential fatty acids, such as linoleic acid OMEGA6 and alpha-linolenic acid OMEGA3, must be obtained from the diet as they cannot be synthesized by the human body. They are crucial for membrane integrity, eicosanoid production, and brain development. Cis versus trans fatty acids. Naturally occurring unsaturated fats usually have cis configuration, which introduces a kink and increases fluidity. Trans fats, produced industrially by hydrogenation, are linked to cardiovascular disease due to the rigid structure and poor metabolism. Membrane dynamics and lipids. The fluid mosaic model describes the cell membrane as a fluid lipid bilayer with embedded proteins. Membrane fluidity depends on the fatty acid composition and cholesterol content. Lipids also contribute to lipid rafts, specialized microdomains involved in signaling and protein sorting. Clinical relevance. Hyperlipidemia, elevated lipid levels in blood, linked to atherosclerosis. Lipid storage diseases, e.g., Tay-Sachs disease, caused by accumulation of glycolipids due to enzymatic deficiencies. Obesity and metabolic syndrome. Excessive triglyceride storage leads to insulin resistance, inflammation, and cardiovascular complications. Steroid hormone disorders. Deficiency or excessive lipid-derived hormones leads to disorders like Cushing's syndrome, Addison's disease, or androgen insensitivity. Previous year questions PYQs with answers 1. CSIR Net Deck 2017. Which of the following is the most amphipathic molecule in a biological membrane? A. Cholesterol. B. Triacylglycerol. C. Phospholipid. D. Fatty acid. Answer. C. Phospholipid. 2. CSIR Net June 2016. Which of the following is a precursor of prostaglandins? A. Linolenic acid. B. Arachidonic acid. C. Stearic acid. D. Palmitic acid. Answer. B. Arachidonic acid. 3. CSIR Net June 2013. Which of the following lipids is not a constituent of cell membranes? A. Cholesterol. B. Sphingomyelin. C. Triacylglycerol. D. Phosphatidylcholine. Answer. C. Triacylglycerol. Let me know if you want the next lecture on proteins or nucleic acids in the same detailed format. You said, proteins, ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on Unit, molecules and their interaction relevant to biology. Topic, composition, structure and function of biomolecules, proteins. Proteins are the most versatile and abundant macromolecules in living systems, performing a vast range of biological functions including catalysis, enzymes, structural support, transport, signaling, immune response, movement, and gene regulation. They are linear polymers of amino acids joined by peptide bonds, and their specific sequence, structure, and folding pattern determine their unique biological functions. Unlike lipids or carbohydrates, proteins exhibit a hierarchical structural complexity, from primary to quaternary structure, governed by both covalent and non-covalent interactions. 1. Amino acids, building blocks of proteins. Proteins are composed of 20 standard amino acids, each with a central alpha carbon bonded to a carboxyl group, amino group, hydrogen, and a variable R group. Based on the R group, amino acids are categorized as nonpolar hydrophobic, e.g., leucine, valine, phenylalanine. Polar uncharged, e.g., serine, threonine. Positively charged basic, e.g., lysine, arginine. Negatively charged acidic, e.g., aspartate, glutamate. At physiological pH approximately 7.4, amino acids exist as switterions. Their side chains influence solubility, reactivity, and folding behavior. 2. Peptide bond formation. Amino acids are linked by peptide bonds, formed via condensation reactions between the carboxyl group of one amino acid and the amino group of another. The peptide bond is planar and rigid, with partial double bond character due to resonance. The resulting polymer is a polypeptide chain, with an N-terminus amino end and C-terminus carboxyl end. 3. Hierarchical structure of proteins. A. Primary structure. The linear sequence of amino acids in a polypeptide chain, determined by the gene encoding the protein. The sequence dictates the structure and function of the protein. Mutations at this level can lead to diseases, e.g., sickle cell anemia due to E-right pointing arrow V mutation in beta globin. B. Secondary structure. Regular repeating structure stabilized by hydrogen bonds between backbone atoms. Alpha helix, right-handed coil stabilized by H bonds between every fourth residue. Beta sheet, formed by H bonding between beta strands, can be parallel or antiparallel. Turns and loops, non-repetitive, flexible regions that connect secondary structures. C. Tertiary structure. The 3D conformation of a single polypeptide chain, determined by interactions among side chains, R groups. Hydrophobic interactions, drive folding into globular forms. Disulfide bonds, covalent link between cysteines. Ionic interactions, salt bridges, H bonding, van der Waals forces. Folding is spontaneous but may require chaperones, e.g., HSP70, grows to prevent misfolding or aggregation. D. Quaternary structure. Some proteins are multimeric, composed of 2 or more polypeptide subunits. Subunits can be identical or different, e.g., hemoglobin equals 2 alpha plus 2 beta. Quaternary structures are stabilized by non-covalent interactions and sometimes disulfide bonds. 4. Protein classification. Fibrous proteins, long, insoluble, structural, e.g., collagen, keratin. Globular proteins, compact, soluble, functional, e.g., enzymes, hemoglobin. Membrane proteins, embedded in or associated with cell membranes, e.g., ion channels, receptors. 5. Protein functions. Catalysis, enzymes like DNA polymerase, kinases, proteases. Transport, hemoglobin, transferrin. Structural support, actin, tubulin, collagen. Defense, antibodies, complement proteins. Regulation, hormones, insulin, transcription factors. Movement, myosin, kinesin, dynein. Signaling, receptors, GPCRs, cytokines, kinases. Signaling, receptors, GPCRs, cytokines, kinases. 6. Protein folding and denaturation. Correct folding is essential for function. Denaturation disrupts secondary and tertiary structures due to heat, pH extremes, urea, or detergents, but primary structure remains intact. Misfolded proteins can lead to diseases such as Alzheimer's, amyloid beta aggregation, Parkinson's, alpha-synuclein aggregation, prion diseases, infectious misfolded proteins. 7. Protein purification and characterization techniques: chromatography, ion exchange, size exclusion, affinity. Electrophoresis. SDS page separates proteins by size. Western blotting, detection of specific proteins using antibodies. Mass spectrometry, determines molecular weight and sequence. X-ray crystallography and NMR, for 3D structure determination. 8. Protein evolution and domains. Proteins evolve via gene duplication, divergence, and recombination of functional domains, which are structurally and functionally independent units. Domains often recur in different proteins, e.g., SH2, zinc fingers, suggesting modular evolution. Protein function can often be inferred from domain architecture. 9. Post-translational modifications, PTMs. PTMs add functional diversity by chemically modifying amino acids after translation. Examples. Phosphorylation SR, THR, TR regulates activity. Glycosylation, affects stability and recognition. Ubiquitination, marks for degradation. Acetylation, methylation, lipidation, alter protein localization function. Previous year questions PYQs with answers 1. CSIR Net June 2014. Which one of the following amino acids is most likely to be found in the core of a globular protein? A. Serine. B. Valine. C. Aspartic acid. D. Lysine. Answer. B. Valine. Hydrophobic residues like valine are typically buried in the core of folded globular proteins. 2. CSIR Net Deck 2016. Which statement is true about beta sheet structures in proteins? A. Hydrogen bonds form between R groups. B. Parallel beta sheets are more stable than antiparallel. C. Hydrogen bonds form between backbone atoms of polypeptides. D. Peptide bonds are involved in ionic interactions. Answer. C. Hydrogen bonds form between backbone atoms of polypeptides. 3. CSIR Net June 2013. Which technique is most appropriate to determine the 3D structure of a protein? A. ELISA. B. X-ray crystallography. C. SDS page. D. Northern blot. Answer. B. X-ray crystallography. Let me know if you'd like the next lecture on nucleic acids or another unit. You said, nucleic acids. ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on Unit, molecules and their interaction relevant to biology. Topic, composition, structure and function of biomolecules, nucleic acids. Nucleic acids are the hereditary molecules of life that store, transmit, and sometimes catalyze the expression of genetic information. There are two types, DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. These are linear polymers of nucleotides, which are composed of three components, a nitrogenous base, a pentose sugar, and a phosphate group. DNA serves as the long-term storage molecule of genetic information, while RNA has more diverse roles including information transfer, mRNA, catalysis, ribozyme, and structural and regulatory functions, rRNA, tRNA, mRNA. 1. Structure of nucleotides. A nucleotide consists of nitrogenous base, purines, adenine A, guanine G, double ring structures, pyrimidines, cytosine C, thymine T in DNA, uracil U in RNA, single ring, pentose sugar, deoxyribose in DNA, laxo at 2' carbon, ribose in RNA, phosphate group, attached to the 5' carbon of the sugar, responsible for negatively charged backbone and polarity. Nucleotides are joined by phosphodiester bonds between the 3' O of one sugar and the 5' phosphate of the next, forming a sugar-phosphate backbone with 5' to 3' directionality. 2. DNA, structure and features. DNA is a double-stranded, right-handed helix formed by two antiparallel strands held together by complementary base pairing. A pairs with T via 2 hydrogen bonds. G pairs with C via 3 hydrogen bonds. The double helix has major and minor grooves, where proteins interact with specific base sequences. Chargaff's rule, %A equals %T and %G equals %C in double-stranded DNA. This rule does not apply to RNA or single-stranded DNA. Forms of DNA. bDNA, most common, right-handed helix. aDNA, right-handed, more compact, found in dehydrated conditions or in RNA duplexes. bDNA, most common, right-handed helix. aDNA, right-handed, more compact, found in dehydrated conditions or in RNA duplexes. zDNA, left-handed helix, GC-rich regions, implicated in gene regulation. DNA is thermally stable due to hydrogen bonding and base stacking interactions. DNA is thermally stable due to hydrogen bonding and base stacking interactions. Melting temperature Tm depends on GC content. 3. RNA, structure and functions. RNA is usually single-stranded but can form intrastrand base pairs, resulting in stem loop structures. Types and functions. mRNA carries genetic code from DNA to ribosome. tRNA transfers amino acids to ribosomes, has cloverleaf structure. rRNA, structural and catalytic component of ribosomes. snRNA, involved in RNA splicing. mRNA, regulate gene expression post-transcriptionally. Ribozymes, catalytic RNA e.g., self-splicing introns, RNase P. RNA is more chemically reactive and unstable than DNA due to the 2' O group. 4. Central dogma of molecular biology. The flow of genetic information follows. DNA right pointing arrow, RNA right pointing arrow, protein. Replication. DNA makes an exact copy of itself. Transcription. DNA is transcribed into RNA by RNA polymerase. Translation. mRNA is translated into protein by ribosomes. Exceptions exist. Reverse transcription. RNA to DNA in retroviruses via reverse transcriptase. 5. Nucleic acid hybridization and denaturation. Denaturation. Separation of DNA strands by heat or chemical agents. Renaturation. Annealing. Rejoining of complementary strands. Nucleic acid hybridization is the principle behind molecular techniques. Southern blotting. DNA detection. Northern blotting. RNA detection. Microarrays. Global gene expression profiling. PCR. Amplification of DNA using specific primers and Taq polymerase. qPCR. Quantitative real-time PCR. TM. Melting temperature depends on sequence length, GC content, and salt concentration. 6. DNA topology and supercoiling. DNA can exist in relaxed or supercoiled forms. Supercoiling helps in DNA compaction and influences gene expression. Negative supercoiling, common in vivo, promotes strand separation. Topoisomerases, enzymes that relieve supercoiling by breaking and rejoining strands. Type 1, cuts one strand. Type 2, cuts both strands, e.g., DNA gyrase. Antibiotics, e.g., quinolones, and anti-cancer drugs target topoisomerases. 7. DNA packaging in eukaryotes. Eukaryotic DNA wraps around histone proteins to form nucleosomes, the basic unit of chromatin. Core histones, H2A, H2B, H3, H4, 2 of each. Linker histone, H1. Further packaging leads to 30 nanometers fibers, loops, and eventually chromosomes. Epigenetic modifications, e.g., methylation, acetylation, of histones affect gene expression without altering DNA sequence. 8. Functional implications of nucleic acids, genetic storage and inheritance. Mutation and evolution through changes in nucleotide sequence. Gene regulation via RNA interference and epigenetic changes. Catalysis e.g., ribozymes suggesting RNA world hypothesis for origin of life. Previous year questions PYQs with answers 1. CSIR Net June 2015. Which of the following bases is not present in RNA? A. Uracil. B. Thymine. C. Guanine. D. Cytosine. Answer. B. Thymine. In RNA, uracil replaces thymine. 2. CSIR Net Deck 2017. In DNA double helix, a pairs with T and G pairs with C. Which one of the following base compositions is not possible in double-stranded DNA? A equals T. B. G equals CC. A plus G equals T plus CD. A equals G. Answer. D. A equals G. No rule supports equal amounts of purines individually. Only total purines equals total pyrimidines. 3. CSIR Net June 2014. Which of the following statements is correct about RNA? A. RNA is more stable than DNA. B. RNA lacks a hydroxyl group at the 2' position. C. RNA can act as an enzyme. D. RNA forms a double helix with itself. Answer. C. RNA can act as an enzyme. RNA molecules such as ribozymes have catalytic activity. Let me know if you'd like the next topic from this unit, like enzymes or biophysical techniques, in the same detailed format. You said, vitamins. ChatGPT said. Here is your CSIR Net J. A. RNA is more stable than DNA. B. RNA lacks a hydroxyl group at the 2' position. C. RNA can act as an enzyme. D. RNA forms a double helix with itself. Answer. C. RNA can act as an enzyme. RNA molecules such as ribozymes have catalytic activity. Let me know if you'd like the next topic from this unit, like enzymes or biophysical techniques, in the same detailed format. You said, vitamins. ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on Unit, molecules and their interaction relevant to biology topic, vitamins, composition, structure and function. Vitamins are a group of small organic compounds required in minute amounts for the proper functioning of metabolism. Although they do not directly provide energy, they act primarily as coenzymes or precursors of coenzymes, facilitating numerous biochemical reactions in the body. Most vitamins cannot be synthesized by the body in sufficient quantities and must be obtained from the diet. Their deficiency can lead to characteristic diseases, and their roles are central in enzyme catalysis, redox reactions, gene expression, and immunity. Vitamins are broadly classified into 2 categories based on their solubility. Fat-soluble vitamins, A, D, E, and K. Water-soluble vitamins, B complex and C. 1. Fat-soluble vitamins. These are stored in the liver and adipose tissues and do not need to be consumed daily. They are absorbed along with dietary lipids and transported via chylomicrons. Vitamin A, retinoids and carotenoids. Functions, vision as retinal, gene expression, immune function, and epithelial integrity. Sources, liver, dairy products, and carotene-rich vegetables. Deficiency, night blindness, xerophthalmia, keratomalacia. Mechanism, retinoic acid acts as a transcription factor, regulating gene expression involved in cell differentiation. Vitamin D, calciferol. Functions, regulates calcium and phosphate homeostasis, bone mineralization. Sources, synthesized in skin upon UV exposure, fish liver oils, dairy. Deficiency, rickets in children, osteomalacia in adults. Mechanism, acts through nuclear vitamin D receptors to modulate transcription of calcium-transporting proteins. Vitamin E, tocopherols and tocotrienols. Functions, antioxidant, protects membrane lipids from oxidative damage. Sources, vegetable oils, nuts, seeds. Deficiency, hemolytic anemia in newborns, neurological symptoms. Mechanism, scavenges free radicals, preventing peroxidation of polyunsaturated fatty acids. Vitamin K, phylloquinone and menaquinone. Functions, coenzyme for gamma-glutamyl carboxylation of clotting factors 2, 7, X, and X. Sources, green leafy vegetables, gut microbiota synthesis. Deficiency, hemorrhagic disorders, easy bruising. Mechanism, essential for post-translational modification of blood coagulation proteins. 2. Water-soluble vitamins. These are not stored significantly in the body and require regular dietary intake. They generally function as coenzymes in various metabolic pathways. Vitamin B1, thiamine. Coenzyme form, thiamine pyrophosphate TPP. Functions, carbohydrate metabolism, oxidative decarboxylation of alpha-keto acids. Deficiency, beriberi, Wernicke-Korsakoff syndrome, especially in alcoholism. Enzymes, pyruvate dehydrogenase, alpha-ketoglutarate dehydrogenase, transcriptase. Vitamin B2, riboflavin. Coenzymes, FMN and FAD. Functions, redox reactions in electron transport and oxidative metabolism. Deficiency, angular stomatitis, chylosis, glossitis. Enzymes, succinate dehydrogenase, acyl-CoA dehydrogenase. Vitamin B3, niacin. Coenzymes, NAD+, NADP+. Functions, redox reactions, energy metabolism. Functions, redox reactions, energy metabolism. Deficiency, pellagra 4Ds, diarrhea, dermatitis, dementia, death. Can be synthesized from tryptophan. Vitamin B5 pantothenic acid. Component of coenzyme A. Functions, acyl group transfer reactions, fatty acid metabolism. Deficiency, rare, may cause fatigue, GI symptoms, paresthesia. Vitamin B6 pyridoxine, pyridoxal, pyridoxamine. Coenzyme, pyridoxal phosphate PLP. Functions, transamination, decarboxylation, neurotransmitter synthesis. Deficiency, convulsions, peripheral neuropathy. Enzymes, aminotransferases ALT, ast glutamate decarboxylase. Vitamin B7 biotin. Coenzyme for carboxylase enzymes. Functions, fatty acid synthesis, gluconeogenesis. Deficiency, dermatitis, alopecia, caused by raw egg white avidin binds biotin. Vitamin B9 folate. Coenzyme, tetrahydrofolate THF. Functions, 1-carbon transfer in DNA, RNA synthesis. Deficiency, megaloblastic anemia, neural tube defects in fetus. Antagonists, methotrexate inhibits DHFR, blocking THF formation. Vitamin B12 cobalamin. Functions: DNA synthesis, fatty acid and amino acid metabolism. Coenzymes: methylcobalamin, 5-deoxyadenosylcobalamin. Deficiency: pernicious anemia, neuropathy, methylmalonic acidemia. Needs intrinsic factor for absorption in the ileum. Vitamin C, ascorbic acid. Functions: antioxidant, collagen synthesis, iron absorption. Deficiency: scurvy, bleeding gums, joint pain. Mechanism: cofactor for proline and lysine hydroxylation in collagen synthesis. Role of vitamins in enzyme catalysis. Many vitamins act as precursors for coenzymes and prosthetic groups essential for the catalytic activity of enzymes. Deficiency of these vitamins impairs the function of specific enzymes, which manifests in clinical symptoms and metabolic disorders. Clinical and biotechnological applications: vitamin-based supplementation in metabolic disorders. Diagnostic biomarkers for deficiencies, e.g., methylmalonic acid for B12. Folate antagonists, e.g., methotrexate, used in chemotherapy. Vitamin C in antioxidant therapy and wound healing. Previous year questions PYQs with answers 1. CSIR Net Deck 2013. Which one of the following vitamins acts as a coenzyme in carboxylation reactions? A. Biotin. B. Thiamine C, riboflavin D, niacin. Answer: A. Biotin. Biotin is the coenzyme for carboxylases like pyruvate carboxylase and acetyl-CoA carboxylase. 2. CSIR Net June 2015. A deficiency of which vitamin causes megaloblastic anemia? A. Vitamin B1. B. Vitamin B9C, vitamin B6D, vitamin C. Answer: B. Vitamin B9. Folate is essential for nucleotide synthesis. Its deficiency leads to impaired DNA replication and anemia. 3. CSIR Net Deck 2018. Which of the following statements is true about vitamin K? A. It is water soluble. B. It is required for DNA synthesis. C. It is required for gamma carboxylation of clotting factors. D. It acts as an antioxidant. Answer: C. It is required for gamma carboxylation of clotting factors. Vitamin K modifies glutamate residues on clotting proteins to allow calcium binding. Let me know when you're ready for the next topic: enzymes, bioenergetics, or any specific area from this unit. You said. Stabilizing interactions. ChatGPT said. Here is your CSIR Net JRF Life Sciences high quality lecture on Unit: Molecules and their interaction relevant to biology. Topic: Stabilizing interactions. For CSIR Net JRF, advanced level, with PYQs, no tables, maximum detail. Stabilizing interactions are non-covalent forces that play a critical role in maintaining the structure and function of biological macromolecules, including proteins, nucleic acids, and lipid membranes. While covalent bonds establish the primary structure of macromolecules, it is a network of stabilizing interactions that ensure proper folding, three-dimensional conformation, molecular recognition, and dynamic behavior in the biological environment. These interactions are reversible, non-covalent, and relatively weak individually, but in combination they provide significant structural stability and specificity. Their disruption can lead to denaturation, loss of biological activity, and disease. Understanding these interactions is fundamental to the fields of molecular biology, structural biology, drug design, and enzymology. 1. Hydrogen bonds. Hydrogen bonding is one of the most critical stabilizing interactions in biology. It occurs when a hydrogen atom covalently bonded to a highly electronegative atom, like N or O, forms a weak electrostatic interaction with another electronegative atom. In proteins, stabilize alpha helices and beta sheets in secondary structure. In nucleic acids, responsible for base pairing, A equals T with 2 H bonds, G is defined as C with 3 H bonds, maintaining the double helix. In enzymes and receptors, aid in substrate recognition and transition state stabilization. Though individually weak, 1-5 kilocalories per mole, multiple hydrogen bonds can produce substantial stability and specificity. 2. Ionic interactions, salt bridges. These occur between oppositely charged side chains of amino acids, e.g., lysine and glutamate. Salt bridges combine electrostatic attraction with hydrogen bonding. Role in proteins, commonly found in enzyme active sites and protein-protein interfaces. pH sensitivity. These interactions can be disrupted by changes in pH, affecting protein stability. Salt bridges are important in thermostability of proteins, especially in extremophiles where high-temperature environments require enhanced structural rigidity. 3. Hydrophobic interactions. Hydrophobic interactions are entropy-driven forces where nonpolar groups aggregate in aqueous environments to minimize their disruptive effect on water structure. Driving force of protein folding. Nonpolar side chains cluster in the protein core. Membrane formation. Lipid bilayers form due to hydrophobic tails avoiding water. Molecular recognition. Active sites often have hydrophobic patches for ligand binding. Hydrophobic interactions are crucial for tertiary structure and are significantly influenced by temperature and solvent polarity. 4. Van der Waals forces, London dispersion forces. These are transient dipole-induced dipole interactions that occur between all atoms in close proximity. Role in protein packing. Allow tight packing of atoms in the hydrophobic core. Enzyme substrate binding. Ensure snug fit in active sites through shape complementarity. Though individually very weak, 0.5-1 kilocalorie per mole, the cumulative effect of many van der Waals interactions contributes significantly to overall stability. 5. Pi-pi stacking and cation-pi interactions. These interactions occur between aromatic rings or between a cation, like Li+, and an aromatic system, like phenylalanine. In nucleic acids, pi-pi stacking between bases adds to helical stability. In proteins, found in ligand binding sites and protein interfaces. In receptors, important for neurotransmitter recognition, e.g., acetylcholine with aromatic cage in receptor. They play a crucial role in molecular recognition and folding fidelity. 6. Dipole-dipole and dipole-induced dipole interactions. Molecules with permanent dipoles can align in ways that allow favorable interactions. In protein-solvent interactions, help orient polar side chains at the surface. Stabilization of protein folding intermediates, through favorable dipole alignments. These interactions are weaker than hydrogen bonds but stronger than van der Waals forces. 7. Water-mediated interactions. Water molecules often mediate interactions between biomolecules by forming bridging hydrogen bonds. In enzyme active sites, water can assist in proton transfer or ligand positioning. In protein interfaces, water bridges enhance specificity in protein-protein recognition. Ignoring solvent effects can lead to misinterpretation of molecular stability and interaction energies. 8. Disulfide bridge is technically covalent, but often mentioned in stabilizing context. These are covalent bonds between two cysteine residues forming cysteine. While not a non-covalent interaction, disulfide bonds provide redox-regulated stability to extracellular proteins. Example, immunoglobulins, insulin, and keratin contain disulfide bridges. Breaking and reforming, involved in protein folding pathways. Biological significance. Protein folding, stabilizing interactions guide and maintain the native conformation. Molecular recognition, ensure specificity in ligand binding, enzyme catalysis, and signal transduction. Structural integrity, maintain double helix in DNA, globular conformation in proteins, and bilayer formation in membranes. Evolutionary advantage, organisms have evolved to fine-tune these interactions for optimal function under different conditions, e.g., thermophilic versus mesophilic proteins. Previous year questions PYQs with answers 1. CSIR Net Deck 2014. Which of the following interactions predominantly contribute to the stability of protein tertiary structure? A. Peptide bonds. B. Disulfide bonds only. C. Hydrophobic interactions. D. Hydrogen bonds only. Answer C. Hydrophobic interactions. Hydrophobic collapse is the major driving force for tertiary structure formation. 2. CSIR Net June 2019. Which of the following contributes the most to the specificity of interaction between a ligand and a protein receptor? A. Van der Waals forces. B. Hydrophobic effect. C. Hydrogen bonding and electrostatic interactions. D. Covalent bonds. Answer C. Hydrogen bonding and electrostatic interactions. Specificity is primarily governed by directional interactions like H bonds and ionic forces. 3. CSIR Net Deck 2016. The pi-pi stacking interactions play a major role in stabilizing a tertiary structure of proteins. B. Alpha helical structure of protein. C. Base pair stacking in DNA. D. Disulfide linkage in proteins. Answer C. Base pair stacking in DNA. Pi-pi interactions between adjacent bases in DNA provide significant structural stability. Let me know when you're ready for the next lecture on enzymes, bioenergetics, or any other CSIR Net life science unit. I'll keep the same quality and format. You said, Van der Waals. ChatGPT said. Here is your CSIR Net JRF Life Sciences high quality lecture on Unit, Molecules and their interaction relevant to biology topic, Van der Waals forces. Van der Waals forces are weak, non-covalent interactions that occur between molecules as a result of temporary fluctuations in the electron density of atoms. These interactions are ubiquitous in biological systems and play a crucial role in the structure and behavior of molecules, especially in large macromolecules like proteins, nucleic acids, and lipids. Despite being individually weak, typically in the range of 0.5-1 kilocalorie per mole, their cumulative effect contributes significantly to molecular recognition, molecular packing, and the stabilization of biomolecular structures. Van der Waals forces are generally divided into two types. Dispersion forces, London forces. Dipole-dipole interactions. Both of these types are based on electrostatic interactions but differ in the nature of the molecules involved. 1. Dispersion forces, London forces. These are the most common form of Van der Waals forces and occur due to instantaneous dipoles that are created when the electron distribution in an atom or molecule becomes temporarily uneven. This transient dipole can induce a dipole in neighboring molecules, resulting in an attraction between them. Nature, temporary or induced dipoles. Importance. These forces are distance dependent, meaning their strength increases as the interacting molecules approach one another. At very short distances, they are repulsive, but at intermediate distances, they become attractive. In biological systems, dispersion forces are important in the packing of hydrophobic molecules in proteins, where nonpolar amino acid side chains aggregate in the protein core to avoid water molecules, contributing to the protein's tertiary structure. 2. Dipole-dipole interactions. These occur between molecules that have permanent dipoles, where there is a partial positive charge on one end and a partial negative charge on the other. The positive end of one molecule is attracted to the negative end of another molecule. In polar molecules, molecules with significant electronegativity differences, such as water, can engage in dipole-dipole interactions. In proteins and nucleic acids, these interactions are crucial for maintaining the secondary structure of proteins, such as in the formation of alpha helices and beta sheets. In DNA, dipole-dipole interactions can assist in the stabilization of base pairing through hydrogen bonds, where the polar nature of nitrogen and oxygen atoms contributes to the specificity of base pairing, e.g., AT, GC. 3. Role of Van der Waals forces in protein structure. In proteins, Van der Waals forces are essential for the folding and stability of the tertiary structure. While hydrophobic interactions and hydrogen bonds are often emphasized, the role of Van der Waals forces cannot be underestimated. Packing of the hydrophobic core. The hydrophobic side chains of amino acids in the protein core are stabilized by Van der Waals interactions. These interactions help to minimize the exposure of nonpolar residues to the aqueous environment. Protein-protein interaction. In protein-protein interfaces, Van der Waals forces contribute to the tight packing and complementary shape needed for high specificity and binding affinity. Induced fit mechanism. Enzymes often undergo conformational changes upon substrate binding, and Van der Waals forces are involved in fine-tuning the enzyme-substrate interaction by facilitating close contact. 4. Van der Waals forces in nucleic acids. Van der Waals interactions play an important role in stabilizing the double helix structure of DNA. While hydrogen bonds between complementary base pairs are crucial for base pairing specificity, pi-pi stacking interactions between the aromatic rings of adjacent bases further stabilize the DNA helix. Base pairing. The interaction of bases through Van der Waals forces, along with hydrogen bonds, provides the structural integrity of the double helix. DNA packing. In chromatin, Van der Waals interactions contribute to the compaction and folding of DNA, interacting with proteins. While hydrogen bonds between complementary base pairs are crucial for base pairing specificity, pi-pi stacking interactions between the aromatic rings of adjacent bases further stabilize the DNA helix. Base pairing. The interaction of bases through Van der Waals forces, along with hydrogen bonds, provides the structural integrity of the double helix. DNA packing. In chromatin, Van der Waals interactions contribute to the compaction and folding of DNA, interacting with proteins to form the nucleosome structure. 5. Van der Waals forces in membrane formation. In biological membranes, lipid bilayers are stabilized by hydrophobic interactions, but Van der Waals forces also play a crucial role in stabilizing the hydrophobic core of the lipid bilayer. These forces are responsible for lipid packing. Lipid molecules arrange themselves in a bilayer with their hydrophobic tails interacting via van der Waals forces, forming a stable membrane structure. Membrane fluidity. The nature of van der Waals interactions between lipid tails can influence the fluidity and permeability of the membrane. 6. Van der Waals forces in drug design. The understanding of van der Waals interactions is critical in the field of drug design, especially for the development of small molecules that can bind to enzyme active sites or receptors. The affinity of a ligand for a target is often determined by its ability to form van der Waals interactions in addition to hydrogen bonds and electrostatic forces. Ligand binding. Drugs can be designed to interact specifically with the van der Waals surface of a receptor or enzyme, ensuring a high degree of selectivity. Molecular docking. In computational drug design, van der Waals interactions are an essential part of the algorithms used to predict how small molecules bind to their targets. Previous year questions PYQs with answers 1. CSIR NET Deck 2012. Which of the following interactions is most responsible for the stabilizing of protein tertiary structure? A. Ionic bonds. B. Hydrogen bonds. C. Van der Waals interactions. D. Covalent bonds. Answer. C. Van der Waals interactions. Van der Waals interactions, together with hydrophobic interactions, contribute to the packing and stability of protein tertiary structure. 2. CSIR NET June 2014. In a DNA molecule, which of the following interactions plays a role in stabilizing the double helix? A. Hydrophobic interactions between phosphate groups. B. Pi-pi stacking between adjacent purine and pyrimidine bases. C. Covalent bonds between sugar and phosphate groups. D. Ionic interactions between complementary. Stabilizing interactions. ChatGPT said. Here is your CSIR NET JRF Life Sciences High Quality Lecture on Unit, Molecules and Interaction Relevant to Biology Topic, Stabilizing Interactions. For CSIR NET JRF, Advanced Level, with PYQs, no tables, maximum detail. Stabilizing interactions are non-covalent forces that play a critical role in maintaining the structure and function of biological macromolecules, including proteins, nucleic acids, and lipid membranes. While covalent bonds establish the primary structure of macromolecules, it is a network of stabilizing interactions that ensure proper folding, three-dimensional conformation, molecular recognition, and dynamic behavior in the biological environment. These interactions are reversible, non-covalent, and relatively weak individually, but in combination they provide significant structural stability and specificity. Their disruption can lead to denaturation, loss of biological activity, and disease. Understanding these interactions is fundamental to the fields of molecular biology, structural biology, drug design, and enzymology. 1. Hydrogen bonds. Hydrogen bonding is one of the most critical stabilizing interactions in biology. It occurs when a hydrogen atom covalently bonded to a highly electronegative atom, like NO, forms a weak electrostatic interaction with another electronegative atom. In proteins, stabilize alpha helices and beta sheets in secondary structure. In nucleic acids, responsible for base pairing, N equals T with 2 H bonds, G is defined as C with 3 H bonds, maintaining the double helix. In enzymes and receptors, aid in substrate recognition and transition state stabilization. Though individually weak, 1 minus 5 kilocalories per mole, multiple hydrogen bonds can produce substantial stability and specificity. 2. Ionic interactions, salt bridges. These occur between oppositely charged side chains of amino acids, e.g., lysine and glutamate. Salt bridges combine electrostatic attraction with hydrogen bonding. Role in proteins, commonly found in enzyme active sites and protein-protein interfaces. pH sensitivity. These interactions can be disrupted by changes in pH, affecting protein stability. Salt bridges are important in thermostability of proteins, especially in extremophiles where high temperature environments require enhanced structural rigidity. 3. Hydrophobic interactions. Hydrophobic interactions are entropy-driven forces where nonpolar groups aggregate in aqueous environments to minimize their disruptive effect on water structure. Driving force of protein folding. Nonpolar side chains cluster in the protein core. Membrane formation. Lipid bilayers form due to hydrophobic tails avoiding water. Molecular recognition. Active sites often have hydrophobic patches for ligand binding. Hydrophobic interactions are crucial for tertiary structure and are significantly influenced by temperature and solvent polarity. 4. Van der Waals forces. London dispersion forces. These are transient dipole-induced dipole interactions that occur between all atoms in close proximity. Role in protein packing. Allow tight packing of atoms in the hydrophobic core. Enzyme substrate binding. Ensure snug fit in active sites through shape complementarity. Though individually very weak, 0.5 minus 1 kilocalorie per mole, the cumulative effect of many van der Waals interactions contributes significantly to overall stability. 5. Pi-pi stacking and cation-pi interactions. These interactions occur between aromatic rings or between a cation, like Li+, and an aromatic system, like phenylalanine. In nucleic acids, pi-pi stacking between bases adds to helical stability. In proteins, found in ligand binding sites and protein interfaces. In receptors, important for neurotransmitter recognition, e.g., acetylcholine with aromatic cage and receptor. They play a crucial role in molecular recognition and folding fidelity. 6. Dipole-dipole and dipole-induced dipole interactions. Molecules with permanent dipoles can align in ways that allow favorable interactions. In protein-solvent interactions, help orient polar side chains at the surface. Stabilization of protein folding intermediates through favorable dipole alignments. These interactions are weaker than hydrogen bonds but stronger than van der Waals forces. 7. Water-mediated interactions. Water molecules often mediate interactions between biomolecules by forming bridging hydrogen bonds. In enzyme active sites, water can assist in proton transfer or ligand positioning. In protein interfaces, water bridges enhance specificity in protein-protein recognition. Ignoring solvent effects can lead to misinterpretation of molecular stability and interaction energies. 8. Disulfide bridges technically covalent, but often mentioned in stabilizing context. These are covalent bonds between two cysteine residues forming cysteine. While not a non-covalent interaction, disulfide bonds provide redox-regulated stability to extracellular proteins. Example: immunoglobulins, insulin, and keratin contain disulfide bridges. Breaking and reforming, involved in protein folding pathways. Biological significance. Protein folding, stabilizing interactions guide and maintain the native conformation. Molecular recognition, ensure specificity in ligand binding, enzyme catalysis, and signal transduction. Structural integrity, maintain double helix in DNA, globular conformation in proteins, and bilayer formation in membranes. Evolutionary advantage, organisms have evolved to fine-tune these interactions for optimal function under different conditions, e.g., thermophilic versus mesophilic proteins. Previous year questions PYQs with answers 1. CSIR Net Deck 2014. Which of the following interactions predominantly contribute to the stability of protein tertiary structure? A. Peptide bonds. B. Disulfide bonds only. C. Hydrophobic interactions. D. Hydrogen bonds only. Answer. C. Hydrophobic interactions. Hydrophobic collapse is a major driving force for tertiary structure formation. 2. CSIR Net June 2019. Which of the following contributes the most to the specificity of interaction between a ligand and a protein receptor? A. Van der Waals forces. B. Hydrophobic effect. C. Hydrogen bonding and electrostatic interactions. D. Covalent bonds. Answer. C. Hydrogen bonding and electrostatic interactions. Specificity is primarily governed by directional interactions like H bonds and ionic forces. 3. CSIR Net Deck 2016. The pi-pi stacking interactions play a major role in stabilizing a tertiary structure of proteins. B. Alpha helical structure of protein. C. Base pair stacking in DNA. D. Disulfide linkage in proteins. Answer. C. Base pair stacking in DNA. Pi-pi interactions between adjacent bases in DNA provide significant structural stability. Let me know when you're ready for the next lecture on enzymes, bioenergetics, or any other CSIR Net life science unit. I'll keep the same quality and format. You said. Van der Waals ChatGPT said. Here is your CSIR Net JRF Life Sciences high quality lecture on unit molecules and their interaction relevant to biology topic. Van der Waals forces. Van der Waals forces are weak, non-covalent interactions that occur between molecules as a result of temporary fluctuations in the electron density of atoms. These interactions are ubiquitous in biological systems and play a crucial role in the structure and behavior of molecules, especially in large macromolecules like proteins, nucleic acids, and lipids. Despite being individually weak, typically in the range of 0.5 minus 1 kilocalorie per mole, their cumulative effect contributes significantly to molecular recognition, molecular packing, and the stabilization of biomolecular structures. Van der Waals forces are generally divided into two types: dispersion forces, London forces, dipole-dipole interactions. Both of these types are based on electrostatic interactions but differ in the nature of the molecules involved. 1. Dispersion forces, London forces. These are the most common form of van der Waals forces and occur due to instantaneous dipoles that are created when the electron distribution in an atom or molecule becomes temporarily uneven. This transient dipole can induce a dipole in neighboring molecules, resulting in an attraction between them. Nature: temporary or induced dipoles. Importance: these forces are distance dependent, meaning their strength increases as the interacting molecules approach one another. At very short distances, they are repulsive, but at intermediate distances, they become attractive. In biological systems, dispersion forces are important in the packing of hydrophobic molecules in proteins, where nonpolar amino acid side chains aggregate in the protein core to avoid water molecules, contributing to the protein's tertiary structure. 2. Dipole-dipole interactions. These occur between molecules that have permanent dipoles, where there is a partial positive charge on one end and a partial negative charge on the other. The positive end of one molecule is attracted to the negative end of another molecule. In polar molecules, molecules with significant electronegativity differences, such as water, can engage in dipole-dipole interactions. In proteins and nucleic acids, these interactions are crucial for maintaining the secondary structure of proteins, such as in the formation of alpha helices and beta sheets. In DNA, dipole-dipole interactions can assist in the stabilization of base pairing through hydrogen bonds, where the polar nature of nitrogen and oxygen atoms contributes to the specificity of base pairing, e.g., AT, GC. 3. Role of van der Waals forces in protein structure. In proteins, van der Waals forces are essential for the folding and stability of the tertiary structure. While hydrophobic interactions and hydrogen bonds are often emphasized, the role of van der Waals forces cannot be underestimated. Packing of the hydrophobic core. The hydrophobic side chains of amino acids in the protein core are stabilized by van der Waals interactions. These interactions help to minimize the exposure of nonpolar residues to the aqueous environment. Protein-protein interaction. In protein-protein interfaces, van der Waals forces contribute to the tight packing and complementary shape needed for high specificity and binding affinity. Induced fit mechanism. Enzymes often undergo conformational changes upon substrate binding, and van der Waals forces are involved in fine-tuning the enzyme-substrate interaction by facilitating close contact. 4. Van der Waals forces in nucleic acids. Van der Waals interactions play an important role in stabilizing the double helix structure of DNA. While hydrogen bonds between complementary base pairs are crucial for base pairing specificity, pi-pi stacking interactions between the aromatic rings of adjacent bases further stabilize the DNA helix. Base pairing. The interaction of bases through van der Waals forces, along with hydrogen bonds, provides the structural integrity of the double helix. DNA packing. In chromatin, van der Waals interactions contribute to the compaction and folding of DNA, interacting with proteins to form the nucleosome structure. 5. Van der Waals forces in membrane formation. In biological membranes, lipid bilayers are stabilized by hydrophobic interactions, but van der Waals forces also play a crucial role in stabilizing the hydrophobic core of the lipid bilayer. These forces are responsible for lipid packing. Lipid molecules arrange themselves in a bilayer with their hydrophobic tails interacting via van der Waals forces, forming a stable membrane structure. Membrane fluidity. The nature of van der Waals interactions between lipid tails can influence the fluidity and permeability of the membrane. 6. Van der Waals forces in drug design. The understanding of van der Waals interactions is critical in the field of drug design, especially for the development of small molecules that can bind to enzyme active sites or receptors. The affinity of a ligand for a target is often determined by its ability to form van der Waals interactions in addition to hydrogen bonds and electrostatic forces. Ligand binding. Drugs can be designed to interact specifically with the van der Waals surface of a receptor or enzyme, ensuring a high degree of selectivity. Molecular docking. In computational drug design, van der Waals interactions are an essential part of the algorithms used to predict how small molecules bind to their targets. Previous year questions PYQs with answers 1. CSIR Net Deck 2012. Which of the following interactions is most responsible for the stabilizing of protein tertiary structure? A. Ionic bonds. B. Hydrogen bonds. C. Van der Waals interactions. D. Covalent bonds. Answer. C. Van der Waals interactions. Van der Waals interactions, together with hydrophobic interactions, contribute to the packing and stability of protein tertiary structure. 2. CSIR Net June 2014. In a DNA molecule, which of the following interactions plays a role in stabilizing the double helix? A. Hydrophobic interactions between phosphate groups. B. Pi-pi stacking between adjacent purine and pyrimidine bases. C. Covalent bonds between sugar and phosphate groups. D. Ionic interactions between complementary base pairs. Answer. B. Pi-pi stacking between adjacent purine and pyrimidine bases. Pi-pi stacking interactions are van der Waals forces between the aromatic rings of adjacent nitrogenous bases in DNA. 3. CSIR Net Deck 2017. Which of the following statements regarding van der Waals forces is false? A. They are strong at very close distances. B. They are involved in molecular recognition. C. They increase in strength with the size of the molecules involved. D. They are electrostatic interactions between permanent dipoles. Answer. D. They are electrostatic interactions between permanent dipoles. Van der Waals forces include both temporary dipoles, London dispersion, and permanent dipoles, dipole-dipole interactions, but they are generally weak and short-range interactions. Let me know if you need further elaboration or the next lecture on enzyme mechanisms, bioenergetics, or any other topic from CSIR Net Life Sciences. You said. Electrostatic. ChatGPT said. Here is your CSIR Net JRF. Here is your CSIR Net JRF Life Sciences high quality lecture on 5. Van der Waals forces in membrane formation. In biological membranes, lipid bilayers are stabilized by hydrophobic interactions, but van der Waals forces also play a crucial role in stabilizing the hydrophobic core of the lipid bilayer. These forces are responsible for lipid packing. Lipid molecules arrange themselves in a bilayer with their hydrophobic tails interacting via van der Waals forces, forming a stable membrane structure. Membrane fluidity. The nature of van der Waals interactions between